We were studying fungi, viruses, and the remaining classification, and we left the discovery of microorganisms here up here. We left this discovery of microorganisms and went straight to the landmarks of the microbiology history. We were studying the comparison of the microorganisms. Why is it not working? Yeah, the history of microbiology with the history of the world, the major events occurring at the time, the major occurring happening in the microbiology. So we were here at the American Civil War, and after that, Pasteur disproves the spontaneous generation and the Dostoevsky's Crime and Punishment novel, or you can say book, and then the Lister publishes on antiseptic therapy in 1867. So we are going to move further from that, and it's actually a lot. I'll show you how much. See, this is a book, and these are the more further, and here it is ending. It is ending on 2003 to 2005. Since this book is the 7th edition of the Prescott's Microbiology, so that's why I think it is ending on 2005. Well, we will deal with it till now. We can always study the microbiology in the recent years, and we can't even keep up with the microbiology discoveries happening after the 2005. So anyway, we were here. We were here in the 1870 to 71. So wait, okay, here 1870 to 71, Franco-German War. By the time this France and German war was happening, Koch demonstrated that anthrax caused the Bacillus anthracis. Anthrax was the microorganism, anthrax was the disease, and it was caused by the Bacillus anthracis. Koch demonstrated that the microorganisms actually causing this disease, Bacillus anthracis. And let me show you. Wait, aren't we missing something? Yeah, no. Why are we directly at the Koch postulates then? Okay, whatever. Koch demonstrated anthrax caused by the Bacillus anthracis. So I want to go and see what actually is this Bacillus anthracis. Anthracis is. I think as a microbiologist, we all should try to know what these names means. Is a gram-positive and rod-shaped bacterium, and is a deadly disease to livestock and occasionally to humans. See, a deadly disease, and it is only permanent pathogen within the genus Bacillus. It is the only permanent. Bacillus are innocent bacteria, while this one just ruins the image of the nice bacteria Bacillus, and it is the permanent pathogen. Its infection is type of zoonosis. Yeah, they are mainly focused on animal attack rather than human attack. By the way, what they actually do what does they do to human? Okay, your body being unable to respond to infection, normally leading to damage of the multiple organ system. Sepsis. Okay, multiple damage, breakage occur in your body, all at a time, and all is this because of this disease, this Bacillus anthracis. Then by the time he was demonstrating that microorganism causes the disease Bacillus anthracis, Oh my gosh. The disease, yeah, anthrax is caused by Bacillus anthracis. The Bell was inventing the telephone. By the way, this telephone looks like the telephone that the children uses, two cups and a wire attached to it. So our early telephones was looking same like that. I'll show you. See, it is the first telephone, a telecommunication company that was founded in Boston in 1877. See the date by Alexander Graham Bell. Let's compare it with our PDF. In our PDF it is written as 1876 and in Google it is 1876. Well, it's not by far that wrong. So here I was trying to show you the picture. This is the telephone, early telephone. So I think there are these two wires, see, and the wire is connected to one of them and then the other of them, and one side of the person listens to another side. Here we can see how it works well. There is a certain example. See, one is for listening and one is for talking. So it's just one way micro telephone. I don't think it works two ways. Or maybe if one is talking, other can only listen. So in 1876, Bell invented the telephone, and by the time we were discovering the microorganisms that were causing diseases. And when in 1879, Edison's first light bulb. Well, if you guys watch the anime Dr. Stone, there is a mention of that Edison's light bulb in there, and he actually recreated the light bulb in original form. It just needs a glass, a vacuum, and a wire, and a little bit electricity. Well, not, yeah, a little bit, and that's all. So the Edison's light bulb looked like this. Yeah, it's kind of elongated and not looks like, doesn't look like ours. See, now this shape is not even elongated, but if you focus on the points, this part is always elongated in the past bulbs. Wait, where did we go? See, this parts, these parts, these parts. These are also known as filament bulb, and if you look at the lighting, is the yellow light. So this just shows that the wire that we're using were not the tangent one. There is a metal used, what do you call it, the one that is also used in the chips of computers. Anyway, in 1879, Edison's first light bulb. The first 1879 was the first time that our earth was getting ready to the light pollution that we are facing these days, and we can't even see stars because of all of these stars at the bottom of the, at the earth. Anyway, in 1880, Laveran discovered the plasmodium, the cause of malaria, just as Koch discovered that Bacillus anthracis was the cause of anthrax. The same case with the, wait, what is happening? What is happening? Yeah, here we are. The cause of malaria. In 1881, the Koch cultures bacteria on gelatin. Okay, for now they were just discovering that the causes of the diseases were microorganisms, but now in 1881, Koch's cultured the bacteria on the gelatin. I think it is for the first time a microorganism was cultured. And the first culture media is the material where the microorganisms can grow. And the first culture media was the gelatin. I think gelatin does have carbohydrates in it. That's why it was easier for bacteria to grow in it, since all they need is glucose for growth. well, the simpler ones. Gelatin nutrients, should we call that? Yes, so we can see we are not looking at the images. So total fat, cholesterol, total carbohydrate is 14 gram, protein is 1.2 grams, sodium is 75 milligram, and I think this is more than enough, since just as I told you that they need carbohydrates for simple, for simple term, and a little bit protein is also good. So he it was easy, but it was a first idea to even grow the microorganism on something. When Pasteur developed the anthrax vaccine, as soon as he grew the bacteria, he started experimenting on them, and then he soon developed the anthrax vaccine. This was in 1881. In 1884, you guys can see it. In 1884, Mark Twain's Adventure of Huckleberry Finn. Adventure. It is the book Adventures of Huckleberry Finn. It's the book or novel about a boy and a slave or things like that. Wait, I'll just search the description. But wait, Twain's Adventure. Yeah, here it is. I don't know why it was famous, but it was famous at that time. That's why it's shown in the map. Even the movie is from. See, look, guys, watch it. By Mark Twain. It must be good enough for it to be so popular at that time in 1884. It's a novel by American author by Mark, published in the United Kingdom. What is it about? It is told from the point of view of Huck Finn, a barely literate teen who fakes his own death to escape his abusive, drunken father. He encounters a runaway slave named Jim, and the two embark on a rough journey down. It's interesting. Well, here is the movie. You guys can watch it. It's not advertisement or something. I don't even know what this story is about. But well, let's search it. Why was it famous? I think the question should be this. American classic. It is considered as a classic of American realism and is often named among the great American novels. Well, who cares? Let's see. It's just a popular. You don't know when humans like things. So anyway, when the novel was rising, at that time, Coach was stuck in the laboratory and was experimenting with microorganisms. So in 1882, he discovered the Mycobacterium tuberculosis. Okay, another bacteria. Now then, in 1884, Koch's postulates was published. Mechnikov described the phagocytosis, and autoclave developed, and gram staining developed. Guys, as a microbiologist, first you need to know what Koch's postulates are, and Mechnikov's what is phagocytosis, or what is autoclave, and what is gram staining. These are the basics of microbiology. So I'll go through it one by one. There are four Koch's postulates, and the postulates is about the disease, infectious diseases. So in Koch's postulate, he said that the first point is there are 4 points. In first point, he said that the infectious organism must have microorganisms in it. See, presence of microorganisms in the diseased individual, but not in healthy individual. Of course, healthy individual must not have the microorganisms that are found in the diseased individual. The second point was the isolation and culture. We should or we can culture these microorganisms in a pure culture, and then after we inject them into unhealthy animals, we can infect them. So a healthy individual must develop the disease after being infected with cultured microorganisms. Like it is mandatory for these microorganisms to cause disease in healthy individual. And after you and the microorganisms must be re-isolated from the infected individual to be identical to the original microorganism. So it is in summary, it is saying that we should take the microorganisms from the diseased animal, and then we should culture it, and then we inject into healthy animal, and then we re-isolate it from the healthy individual and compare it with the original one, and the result must be the same, means they must resemble each other. So these are core postulate. It is, as you can see from the history, he was searching or researching on the microorganism. So he, I think, postulates he must have observed these patterns when he was studying the microorganisms that were causing diseases. So he just made the postulates of it. Now the another scientist, Mechnikov, described the phagocytosis. It is simple phagocytosis. Cytos means, I think cyto word is derived from cells. Phago is eating. So it is the eating of things by the cell. So when the cell in what do you call ingest outer materials or foreign materials and dissolve it in its own and digest it, it's called as phagocytosis. Wait, let's read the correct definition. Wait, not pathogen. Phagocytosis is a cellular process where a cell engulfs and destroys large particles such as microorganisms, foreign substances, dead or damaged cells. It even eats its own cells, and they are larger cells. The phagocytosis is usually done by the macrophages, the immune cells present in the body. See? Wait, I don't know if you can see it. But see, this is the cell. The pink thing is the foreign material. It is engulfed, and after engulfing it destroys it completely. It is blurry so we can see it, so let's leave it at that. So in short, it eats it and destroys it. And that's the process of phagocytosis, just as our digestive system works. Consider your whole body as a cell, and the process you are doing is the phagocytosis. So it is just that. Then after that, this was the autoclave. So autoclave was developed. What was the first autoclave looked like? So the first autoclave looked like the pressure cooker, right? And these days the autoclave looks like the square machines. And yeah, the previous auto machines, autoclaves were simple and they were sealed completely and they didn't have much options as the autoclaves these days are. See, this is the autoclave of the present day, this one, and this one was the of previous days. This one's of previous days. I didn't mean to click it, but whatever. Anyway, wait, I can't even see the comments even though there are none. Okay, so we were where? First motor vehicle by Denter, 1885 to 1886. And this, okay, we left the gram staining. Remember, gram staining is done to differentiate between gram positive microorganism bacteria and gram negative bacteria. As we are going through it, I'll tell you what gram. There are two types of bacteria, and the differentiation is done because of the cell wall. The bacterial cell wall is of two types. One is known as gram positive, one is known as gram negative. The gram word came from the scientist, okay, so don't take it seriously. And as you can see here, wait, This is not the clear one. This one's the clear one. Let's look at that. Okay, so you can move to the side. Here. So if you see, the gram-positive bacteria has peptidoglycan layer. This is the cell membrane, right? And the cell wall is made up of peptidoglycan. Peptido means peptides, glycan means glucose. So the gram-positive only have one type of, cell wall, one type of barrier after the plasma membrane, while in gram-negative, it is sandwiched between two lipid bilayers. So one lipid bilayer is the plasma membrane, while the other one is even outside that peptidoglycan, which was homologous among the gram-positive, while it is smaller in size in gram-negative. But if you see, you must think that the gram-positive is the strengthened one and the strong one, but no. The gram-negative bacteria cell walls are more stronger and more resistant than the gram-positive, since they have two double types of protections against them. So we were talking about gram staining. Staining comes from coloring. So when we use the colorings to see the microorganisms under the microscope, right? So we need some coloring since they are, originally kind of invisible. So when we stain it, these cell walls take different colors. One retains the purple color while one cannot retain the purple color after we wash it. This one loses this outer layer, and so the purple color is gone with it. So we use another type of stain. This is a pink type of stain. This one cannot no longer take the pink stain because this is already colored with blue ones or purple one. But this can take pink since already washed the outer covering. After we wash it and this takes the pink color, we can differentiate that this one is the gram negative one and that one is the gram positive one, right? What am I doing here? See, so the purple one is the gram positive and the pink one is the gram negative. So this is very important in differentiating between the gram positive and gram negative bacteria, this gram staining technique. And what is the history of gram staining technique? As you can see here, the gram staining was around 1884. See, so these are the basics of microbiology and they all occurred at the same time. Now the 1885 to 1860, comparing with the rest of the world, while they were providing the basics of microbiology, the rest of the world was creating motor vehicles. Motor vehicles, by the way, the first motor vehicle is kind of interesting. Motor vehicle of 18, what was it? 25? 1844? I don't know. Was it 45? Oh gosh. Here it is. Yeah, it kind of looked like this. So that's that. Anyway, then 1885, again posture came, and he developed the rabies vaccine, and Escherichia discovered the Escherichia coli. So this was the age where different microorganisms were, cultured and were discovered and were observed. Since I think Koch provided this culturing method, so more and more scientists started to discover more and more types of organisms. And after observing them, they started producing vaccines or providing vaccine. By the way, rabies is not a bacteria. It's not caused by the bacteria. It is caused by the viruses. So I think he did a very good job in providing the vaccine. Then the 1887, wait, you guys can see it. Now the 1887, Richard Julius Petri developed Petri dish. Okay, Petri dish. Why the Petri? It is a scientist named Petri, so they named it Petri, and it is I don't think there was anything to develop in petri dish. Just take a plate, a transparent plate, and then that becomes the petri dish. I don't know why this is just such a big thing. We all know we use the transparent dishes type things in the labs, but why does it go towards the Richard Julius specifically? Is it the glass era or something? I don't know the importance of this. I know it's a very basic thing, but why only this scientist? Multiple must have used it, or was Richard Julius the first one to use petri dish? Who knows? Then the 1888 Hertz discovered the radio waves. Okay, I don't know how he discovered the radio waves, but in one way or another he discovered it. So Hertz discovered radio waves. How? I should have written how first. Okay, so this is it. So he connected the battery to the wires, and then he produced the sparks, and then there was the the signal collector or things like that. Wait, I think we should read the Google here. By using an induction coil to generate high voltage sparks across a spark gap, which created electromagnetic waves that could be detected by a separate receiving antenna with a small spark gap, providing these waves existed and could travel through space, thus confirming the James Clerk Maxwell's theory of electromagnetic radiation. He was able to measure their wavelength and okay, so the physics wasn't that laid back and had the technologies of measuring things like that. So it's understandable. While in 1887 to 1890, what was happening in our microbiology world? The Winogradsky studied the sulfur and nitrifying bacteria. And nitrifying bacteria are the bacteria that converts ammonia into nitrates and nitrites. Only plants can only use nitrates and nitrites forms of nitrogen. It cannot use the ammonia or direct the nitrogen gas present in the atmosphere. So they were, I think this guy was in environmental biology or something, or environmental studies. So he was studying these kinds of bacteria, nitrifying bacteria or sulfur bacteria. They have major roles in the environment. Okay, so after what happened? So this is 1889, this is 1889. Oklahoma land rush. Okay guys, so I studied this before, but the live went wrong and there was no voice. So I kinda know about these things, that's why I'm going through it this smoothly. So about Oklahoma land rush, I studied that there was a territory in the India, and but it wasn't occupied. I don't know how it works, but it wasn't occupied, and the people decided that they should race to that place and claim this spot. Whoever comes first, whoever came first there, it is their place now. So it actually caused quite a disaster, and but people at that time started having a place and a territory, and it developed into a area territory in Oklahoma. So unassigned land. Wait, let's read it. The Oklahoma land was a historical event where thousands of people raced to claim plots of lands in the unassigned lands of the former territory in Oklahoma, starting at the designated time, usually signaled by the gunshot, with the first to reach a piece of land getting to claim it. See, it was like a game. Whoever win or came first took that place. And this happened most notably in April 22, 1889, where roughly 50,000 people rushed to claim the land, marking the beginning of Oklahoma's settlement as a state. 50,000 people are no joke. That many people rushed to that place to claim that land, so of course it's going to become a state. So anyway, when the major happening of the state creation was happening. Beijing isolated root nodule bacteria. This guy was stuck in the roots of the plants to isolate the bacteria, the invisible microorganisms, while there was a state creation happening. Anyway, this is the wonders of microbiology. Things you can't even see and are present in the roots or under the earth, you are searching for them. So in 1889 he did that. Don't underestimate Beijing. He is going to come forward for a great discovery. So in 1890, Eiffel Tower completed. Okay. So I think at that time Eiffel Tower must have been the longest, the biggest tower of the world, right? Wait, what is the first tower of the world? It must be Eiffel Tower. No wonder it's so famous. What is the first longest tower in the world? What is the first tallest tower? I hope it is Eiffel Tower. What? First, what was? I think it should be was. Otherwise, they are not understanding it. Burj Khalifa is just, was worse already. Wait, this is not working. We should write, why is Eiffel Tower famous? Yeah. Famous for including world's tallest building. Don't tell me Eiffel Tower is the longest building. Am I an idiot? I didn't know till now. I thought Burj Khalifa was the... Oh my god. Was the tallest building in the world. Okay, when it was built in 1889, the Eiffel Tower was the tallest building in the world, standing at 330 meters tall. It held the title for 41 years until the Empire State Building was built. Okay. See, I told you. It was the world's tallest building in the world at that time. Well, who am I kidding? Anyway, wait, what is the tallest building now? Okay, we need to know, okay? What is the tallest building in the world? What is it? Okay, I wasn't wrong. Burj Khalifa is the tallest. Then the Warsan Merdeka Tower, then the Shanghai Tower, then the Makah Clock Royal Tower, Golden Finance Tower, Pinghe International, Lotte World Tower, One World Trade Center, Tianjin CTF Finance Center, what? Gongzhou Cao Tai Fok Tower, whatever. Wow, what do you call it? The Eiffel Tower is gone. It is just 330 meters, while the tallest one these days is 828. No, sorry. Yeah, not sorry. 828 meters. While at that time Eiffel Tower was the tallest and it was only 330 meters. The Eiffel Tower must be looking dwarf these days. 1891, Behring's antitoxin for the diphtheria and tetanus. Okay, diphtheria is also a disease. Is a disease that diphtheria is caused by microorganisms that cause disease by producing toxin. The same is the case with the tetanus, right? And they both damage nervous system to some degree. Not to some degree, actually every degree. They cause problems for nervous system. And one Behring's was the first to produce antitoxins for them. See, just as antibiotics works against the bacteria and the vaccines work against the virus, antitoxins works against the toxins. So I think this was a big feat done by the one Behring's. Anyway, There, the Eiffel Tower was completed, and there, the one Behring was producing antitoxins for diphtheria and tetanus. Now, the Yellowstone became the first national park. I don't know the importance. I didn't even know the Yellowstone till I told you that the live went wrong, and I am doing it now again. So it's such a beautiful place. I thought I didn't saw it before, but see, I not yellow fever, Yellowstone. Yellow fever is coming. Wait, coming forward afterwards. Yellowstone. You guys will be shocked. You guys must have seen it, or I think the people living in the U.S. would know. See this. This I didn't know this is known as Yellowstone. This is just a hot spring, isn't it? And this is magma lava or something. This is so dangerous, and they say that they made it the national park to preserve it. I don't know for how are they planning to preserve it, but anyway, this became the first national park. There mustn't have been any national parks before that. And with that, in 1894, Kitasato and Yersin discovered the Yersinia pestis. Okay, guys, the black plague that we were studying in the previous slides, it was caused by the Yersinia pest, the same plague that wiped out the 1/3 of the population of the Europe, and after 80 years, even more than that. And it was caused by this tiny microorganism, Yersinia pestis, and it was discovered by Kitasato and Yersin after that many years. That many years. Okay, it's not that many, of course. I don't know. It happened quite early. Anyway, so in 1895, Bordet discovered the complement. Okay, complement is a heat labile component of the immune system, right? And it is known as component